Doctorado en Nutrición y Microbiología del Rumen en la Universidad de Guelph en 1991. Actualmente es investigador principal en Agriculture and Agri-Food Canada. Tiene numerosos estudios en varios temas como microbioma del rumen, fisiología de infecciones digestivas, emisiones de metano, genómica comparativa con enfoque a los factores asociados a los animales superdiseminadores de Escherichia coli o 157H7 y también estudio de metagenómica para evaluar los riesgos a la inocuidad de alimentos en granjas y en plantas procesadoras. Además de las múltiples publicaciones que tiene el doctor, ha recibido varios premios. Le fue otorgada la medalla Queen Elizabeth II por el premio al Servicio Público de Excelencia debido a su contribución científica en Canadá y el premio Macmillan en Agricultura, así como el premio de la Industria Canadiense de la Carne por su desempeño sobresaliente en investigación e innovación. Entonces tenemos varios ponentes de lujo en la tarde de hoy. El doctor va a dar una plática sobre las aplicaciones en inocuidad de alimentos de la secuenciación completa de genomas de bacterias, así como de la metagenómica, que ya les explicaré de qué se trata eso para los que no sepan. Entonces, bienvenido, doctor. Welcome, Dr. McAllister. You're free to start.
Okay. So can you see my presentation? It doesn't look like you can. Can you bring it up on the screen or? I'm not sure how to do. Maybe that'll do it. There you go. Okay. So everybody, you can hear me all right?
Díganle ahí que sí, que siga.
Okay, I'll start if I can, I know you guys can hear me. Everybody wave if they can hear me. Good, okay. So I'm sorry I don't speak Spanish, so that's gonna maybe be a bit of a problem. But I'll do it in English and hopefully I'll go slow enough people can understand what I'm trying to say. So I was asked, thanks a lot for the invitation. Unfortunately, I couldn't make the trip down. I was just in Germany last week, so I'm still recovering from jet lag from there. So I thought it was just more than I could probably handle and still remain conscious. So, but anyway, I'll go ahead and give the presentation. This is an area that we're doing a lot of work in right now. I think Claudia might have mentioned some of the collaborations we have going on. I would probably describe it that probably is the most rapidly advancing area in science across many disciplines. Food safety or pathogens is only one of the areas that this methodology is now being employed. I also want to recognize my co-author, Dr. Rahat Zahir. She's an integral part of our program here. So we collaborate together a lot on a lot of the work that I'm going to show you. So the original area of focus of science was in the area of genomics, which basically involves sequencing the organism of interest. Since that point in time, it's now expanded into metagenomics, which is very much sequencing DNA that's isolated from various environments. Pretty well any environment of which you can extract DNA from, you can carry out metagenomics. And then more recently, we've also been doing work on metatranscriptomics, which is looking at the genes within the genome that are specifically expressed. So you're getting closer and closer to the point of biological function, which is the translation of the protein and the production of the biologically active protein that ultimately has an effect in biochemical pathways and the responses that we see in the organisms of interest. Obviously, in this presentation, we're interested in the application of these methodologies to the detection and identification of pathogens. So this whole area, as I mentioned, is expanding exponentially, and if anything, the rate of increase, or that rate of exponential increase, is increasing even at a more rapid rate. This goes back a long time, but basically, if you look at the major advancements that have taken place in this area, even those were not that far in the distance past. So 1977, first use of ribosomal RNA, DNA as a method of marker for taxonomy of organisms. The first microbial community studies occurred in 1990, so a little over almost 30 years ago. But basically at that point they were using libraries, which would be considered an archaic way to do that today. In 1999 we had our first metagenomic studies, or proposal of the term metagenomics, and the first next generation sequencing machine was released by Roche in 2005. So that's not really that long ago. The Human Microbiome Project was first published only 10 years ago. In 2011, we had the release of the first long sequencing technology in the form of the PacBio instrumentation, which is only a few years ago. 2015, we had Ocean Sampling Day, where they were collecting samples throughout the globe in the oceanic areas to look at the microorganisms that reside in those environments. 2014, we just had the second long generation sequencing technology release, the nanopore system. So this whole area continues to expand. You can see the number of publications in relation to that area and of metagenomics and how much that has increased in the last few years. Why has this come about? Well, basically it's because of the huge advancements that we have had in sequencing technology, where we started out with the Sanger sequencing system, which was quite expensive, labor intensive, and took a long time to answer any of the genomic questions we were trying to approach. Roche came out with the 454, like I mentioned earlier. That technology would be considered antiquated now as well. And now we're moving into even more advanced technologies like the MinION, which is really now, it was released in 2014. It takes a few years for those to get into more mainstream. Often those new technologies, those new sequencing methodologies, when they're released, have fairly high error rates, like around 18% or so, but they still prove useful when they're coupled with more reliable sequencing technologies like the Illumina, and we sequence the same DNA using both of those methodologies. We can gain even more information than with either alone. And we'll see those same technologies, even the long-read technologies, like for example the PacBio, the error rate associated with that instrumentation has dropped dramatically. So really now sequencing capacity is not the limitation to this work. We can sequence an awful lot of DNA in a short period of time. So just to mention, the nanopore was just MinION. We're just starting to work with this now, measuring up to 250 to 300 kilobases. This has a big implications because it dramatically reduces the amount of computational power you need to try to assemble genomes or try to assemble the material, because the read lengths are so much longer than what they are with the other sequencing methodologies. Where the bottleneck is now emerging is in the whole amount of data that's being generated by these methodologies. So when we do shotgun metagenomic sequencing, which basically means sequencing all the DNA within the sample that we've extracted it from, we are basically then covering all of the metabolic activities associated with that environment. And frequently, we're only interested in one particular trait, for example, like genes associated with virulence or genes associated with antimicrobial resistance. But all of the genes associated with things like carbohydrate metabolism, lipid metabolism, protein metabolism, all of those genes are also being sequenced at the same time. So I would venture to guess that probably if you look at a shotgun metagenomic data set, there probably is not a single data set out there that's been completely mined for all of the information it contains. And so we end up with requiring fairly sophisticated bioinformatic and computational resources in order to assemble and really derive all the information from that data. And that's really then been associated with the sequencing as well as the cost per megabase pair. So if you look at, like in September 01, which is not that long ago, we were looking at about $5,000 per megabase. And now we're down to, and this is 2015, it's even cheaper than this now, it's more down to about 9 cents. It was down to about 14 cents. So if you do a comparison of that in my field, so I do a lot of work in rumen microbiology as well, and I was involved in the first Sanger sequencing programs that sequenced the first bacteria out of the rumen. The cost associated with that program was about $3 million to sequence one bacteria, and it took about 5 years to complete that project. Now we can sequence an individual bacteria and assemble its genome in less than a week for about $200. So the cost has dropped that much. And really that's what's making the difference in where we're going now. So the majority of the work that's been done to date has really been focusing on 16S, RNA, or in case of eukaryotics, 18S RNA, or the ITS region for fungi. Basically that describes who is there, who is in that microbial community, but it does not describe what they're doing. And so often, microbes or bacteria have this amazing ability to enter into this state of dormancy, where they're not, by definition, they're not dead, but as well, they're not metabolically active. And so you could go ahead and sequence that extracted DNA from that environment, and you can identify that those bacteria that are there, but you really don't know what their metabolic contribution is to the community. When we start to get into transcriptomics, then we start to look at gene expression. And by sequencing the messenger RNA, we can then specifically identify those genes that are being expressed. So for example, if we're talking about an antibiotic resistance gene, just because the presence of that gene is in the genome, it doesn't mean that that bacteria necessarily exhibits the antibiotic resistance phenotype. And it would be the same thing for virulence genes. If we talk about virulence genes that are associated with pathogens, just because the virulence gene is present in the genome, it does not mean that the conditions are such that it's being expressed by the pathogen and having the detrimental effects that are associated with that virulence gene. When we start to look at transcriptomics, we then get an idea of whether that gene is expressed and the level of expression that is taking place within the organism as well. And then finally, we've dabbled into the area that's even more complicated, which is getting into the proteomics, because a lot of those genes that are transcribed to messenger RNA may undergo post-translational modification when translation expresses them as proteins. So even at that point, you can have situations where the transcriptomic information is not the be-all and end-all in terms of the metabolic activity that that gene may be having in the form of the protein that is produced. The complication associated with proteomics is the sheer volume of proteins that are expressed within these complex environments and the identification of those individual proteins in a predictive manner. That's still something like, for example, if we extract proteins out of the rumen, a very complex environment like that, we can find that there's a myriad of proteins present, but to really definitively identify each individual protein that's expressed or translated by the microbial population and its function is still something that's, I would say, somewhat beyond our capacity in terms of getting accurate information out of that approach. So just to give you an example then of how we've applied some of these methodologies, I want to emphasize that just because we can do the genomics, the metagenomics, transcriptomics, it doesn't mean we can give up on culturing individual microbes in the lab. Because when we do those types of analyses, we're able to identify or basically characterize the community, but we can't go back and study it after the fact. Often when we're talking about, for example, if we're talking about metagenomics, shotgun metagenomics, and we're looking at AMR determinants, genes that code for antimicrobial resistance, we can identify what antimicrobial resistance determinants are there, but we can't associate them with specific bacteria. In order to do that, we need to get the bacterial isolates and characterize them as well. So this is a project that we're running. Probably Claudia mentioned some of the work that Amelia is doing. Those samples that she's working with come from this project. And what the project is the One Health approach to antimicrobial resistance. We're using E. coli and enterococci as indicator organisms. And then we've collected from the cattle, from the surrounding catch basins, from surface waters, wetlands, and creeks. And then we've also collected samples from wastewater treatment plants from the cities here in Alberta, and also clinical isolates from the hospital communities. And we've compared all of those samples, both from the nature of those bacteria that we're isolating from those various environments, as well as the nature of the DNA that we're finding within those environments as well. Not only from what bacteria that are there, but also what resistance determinants are there, what virulence genes are present, what biocide resistance genes are present, what heavy metal resistance genes are present throughout that continuum. So that's basically the framework we established. It involves collecting from multiple feedlots in Alberta. The wastewater treatment plants are upstream and downstream from the major feedlot producing area here in the province of Alberta. And the human sampling sites are from the major cities that we have, Edmonton and Calgary here. Those are the two largest cities in the province of Alberta. So we're covering all of those samples simultaneously. I know you guys have got big feedlots down in Mexico as well. This is just an aerial view of one of our feedlots here in Alberta. So you can see how the animals are held in the individual, in the pens. Each of those pens will hold anywhere from 150 to 200 head of cattle. And you can also see the catch basins along the side of the feedlot there. Those are meant to capture any of the runoff that comes off of the feedlot during rainfall events. And that material is often irrigated back out onto the pasture land. So we collected samples from the animals within those pens, as well as from the catch basin, and as well from surrounding creeks. You can see a bit of a creek off to the left-hand top corner of this diagram here. We sampled from that creek as well. So you're getting directly where the antimicrobials are administered in the feedlot itself, a distance a little bit further apart in terms of the catch basin, and then further again when we're talking about that creek that's in the vicinity of the feedlot. So we then collected those samples and we looked at the antimicrobial use data, so what antibiotics are being used in the feedlot, and then we collected DNA from those environments, did the sampling as well for sequencing, both using the metagenomic sequencing as well as quantitative PCR. And then we also collected those isolates, the E. coli isolates, both on a genotypic, so identified those species using PCR methodologies, as well as determining their AMR phenotype and genotype. So some of those E. coli that we would be isolating, we deliberately were looking for those E. coli that have the greatest impact on human health. So that's the ESBL, where we enriched for ESBL E. coli. Some of those E. coli that we're isolating could be S-Tech. So they could have the Shiga toxin genes associated with the virulence there as well. So this is just some of the data that we found. This is focusing on enterococci. Enterococci, from a pathogenic perspective, the major issue with them is associated probably with urinary tract infections. Vancomycin-resistant enterococci is probably the most significant pathogen. But in hospitals, they play a major role in the contamination and colonization of catheters. So often catheter-based infections are related to enterococci. And probably where they have the greatest impact in human health is in extended care facilities. They're notorious for causing urinary tract infections in the elderly. And so this just shows the species. Enterococci are much more complicated than the E. coli because, first of all, the plates that we use are less selective in terms of individual species. So we end up selecting, they're very good for selecting the genera, but we get several different species that grow on those plates. And this is just where we use a PCR assay to identify each of these individual species of enterococci. And this just shows you what we find from the collections, various collections that we did using those methodologies. You can see that the majority of the enterococci that were associated with cattle are Enterococcus hirae. As we move to the catch basins, we still have a higher concentration of Enterococcus hirae, but we start to see some of these other species start to emerge. So as you move away from the feedlot, you get other Enterococcus that are contributing to that environment. When you move to the surface water and wetlands in the vicinity of the feedlot, you have even more of those other species that start to move, like environmental Enterococcus like Cassiflavius and Durans. But then when you start to go to the wastewater treatment plant, so now we're starting to deal with material that's closer to the human side of things, we start to see a predominance of Fecalis and Faecium start to emerge in those wastewater treatment plant samples. And then when we go to the human clinical isolates, we almost exclusively find Fecalis and Faecium. So the conclusion from this basically is that the predominant bacteria or enterococci that are in cattle differ substantially from the predominant species of enterococci that we find in humans. So it provides evidence that the linkage between the enterococci in the cattle population are not closely related to the enterococci that cause human health issues in people. Now this is just showing some of the comparative genomics. It shows how obviously when we run those and we do whole genome sequencing on those isolates, they do cluster together, which would not surprisingly, based on the species. So the hirae associated with cattle clusters separately from the Faecium that's associated with human disease. And the same with Fecalis, it also clusters separately. That's a combination of the types of genes that those bacteria harbor. So hirae is adapted to the intestinal tract of cattle. The genes that you need to adapt to that environment differ from the genes that you require to adapt to the intestinal tract or the cause infections in humans. And we find the same thing. You can see that Cassiflavius, Gallinarum, those are the environmental enterococci. Those enterococci require another set of genes that enable them to remain viable within the environment. So a lot of those other intestinal tract based enterococci in the cattle, once they're excreted, those populations start to decline because they're not as competitive out in the open environment as some of these other environmentally adapted species are. Now, it wasn't like we never found Fecalis or Faecium in cattle. We did. It was extraordinarily rare, but we did find some isolates. And even at that point, when we sequence those isolates, so they're the same species, you can see that they still clustered separately for the most part. So the human clinical isolates, as you can see in the bottom part of this figure, tended to cluster together, and the isolates associated with bovine feces also tended to cluster together. So even in the case of the same species, there's differences between those members of that special community that cause disease in humans relative to those that reside in the bovine digestive. More isolates in this case with Fecalis, and you can see the red ones are the ones that are associated with bovine, and the black are the ones that are associated with humans. So they're also clustering separately. So even within the same species, there are differences between the species that cause disease in people and the species that reside in the cattle. Now, occasionally there might be some overlap, and that may very well reflect that the cattle have come in contact with human isolates from humans. And when we talk about the movement of pathogens, we usually talk about the movement from the livestock to the people, but we have to be equally aware that the direction can be the other way, from the people to the livestock. And in the livestock, it's possible that those pathogens are amplified. They may be re-released in the environment and then pose a threat to human health, but that original issue arose in the humans, not in the livestock. So we also can do comparative metagenomics. In this case, what we're doing is we're collecting DNA from these environments. So from the feces, from the catch basin, from the surrounding agricultural lands or the soil, and from the wastewater. And then we do shotgun metagenomic sequencing on all of that DNA. So all of those genes that are in that material, we measure all of those genes that we can relate back to known databases. In the case of resistance, antibiotic resistance, we use a database called MegaRes. And that represents all of the existing defined resistant determinants that are known within the literature and have been published. So there's about 3,000, a little over 3,000 different resistance genes in that database. That database is continuously growing. We just discovered a new antibiotic resistance gene that codes for aminoglycoside resistance. We've added that new discovery to this database. So that database will evolve over time as more sequencing goes on, as more of that DNA that's identified is related to genes coding for antibiotic resistance. Those will be added to this database. So in this study, what we did is we collected DNA from all of these environments, including across the continuum for beef production. So from the ranches, from the feedlots, different areas within the feedlot, during transport of the cattle to the packing plant, and in the slaughter plant as well, including in the retail samples. So basically a cross-continuum analysis of the nature of the DNA within those environments and the antimicrobial resistance genes that it contains. And this is just one figure I want to show you. It represents the number of unique genes that we found in each of those environments. So on the bottom here you see number of unique antimicrobial resistant identified per sample. So the greater the number that is, the more genes that we're finding that are novel within that environment. So if you look at the number of samples, when we're talking about the abattoir, we found no unique antimicrobial resistance genes in that environment. And then if you look at the water, we found fewer, 1 to 20. If you go into the feces, we found more, 21 to 40. But where did we find the most? We found the most in the soil environment. Over 120 new antimicrobial resistance genes determinants were identified in those samples. So the most diversity of antimicrobial resistance was present within the natural soil environment itself, which is not surprising because the majority of antimicrobials that we use in livestock production originated from bacteria within the soil. And so that diversity is still present within those soil samples. The other thing that this tells us is that the techniques that we're using already to control pathogens within abattoirs, whether it be the thermal treatment or the acid washes, etc., those techniques are also effective at preventing bacteria with antimicrobial resistance gene determinants from entering into the food supply as well. And so that's why it sort of has a double effect. It both controls pathogens, but even bacteria that are not pathogens but carry antimicrobial resistance genes, it controls those as well. And that's why those... Okay. So I usually don't practice presentations, but unfortunately, like you guys, I gave one hell of a presentation there, actually, to myself in the room here. I kept going right to the end. So anyway, I'll pick up. I think I finished talking with this slide here, I hope. This is the one I should have left off on. So I think I finished talking about this slide. I'll go on then talking about this. This is metagenomic work that we did where we were isolating from those various environments. So from the cattle feces, from the catch basin, from soil, and from wastewater. And you can see that the microbial populations associated with those environments differ dramatically. The firmicutes predominate within the feces of the animal. The catch basin has a predominance of proteobacteria as well as the soil. And the wastewater has a high level of proteobacteria as well. So you can see that the populations differ depending upon the environment that you're studying. And because the bacteria are related to the factors like antimicrobial resistance and virulence, the types of virulence determinants and antimicrobial resistance genes within those environments change as well. And so this just shows this for the antimicrobial resistance genes. You can see that in the fecal samples associated with the cattle, we have a predominance of tetracycline and macrolide resistance, that's MLS. We use a lot of macrolides and tetracycline in beef cattle diets. When you go to the catch basin, We still have a predominance of tetracycline and macrolids. That's the catch basin directly adjacent to the feedlot that captures the runoff. And then when you go to the soil, you can see you have a very different change in the profile. So this is the surrounding agricultural lands. You have multi-drug resistance associated with the soil bacteria and the glycopeptides. And then if you go to the wastewater treatment plant, you can see that you have an antimicrobial resistance profile that parallels the diversity of antimicrobial use that goes on in humans. Many different types of antibiotics are used, and we pick up ones, for example, the fluoroquinolones, like ciprofloxacin, which we do not detect in the cattle because they don't use a lot of fluoroquinolones in cattle. And then those various environments, when you do principal component analysis, they also cluster separately as an indication of the differences that exist between those environments. We've also done work comparing natural versus conventional feedlot cattle production. So natural lots do not use any antibiotics. Conventional lots do. And you can see that the microbial populations are similar between both of those. So whether or not you use antimicrobials or not doesn't have a major impact on the major populations that are present within those environments. Likewise, it doesn't have a major impact on the types of antimicrobial resistance that's there. You can still see that you have tetracycline and macrolide resistance in the natural lot, but the level of resistance determinants that we're detecting per unit of DNA is lower in the natural relative to the conventional. So if you use antimicrobials, you increase the amount of resistance determinants. If you reduce the use of antimicrobials or even eliminate it, you reduce the presence of resistance determinants, but you do not reduce them to zero. So in this kind of situation, if you reintroduce antimicrobial use into the natural environment, you'd have a really rapid spike in the amount of resistance determinants that we found in relation to tetracycline and macrolides. Although you can establish resistance genes within the population through the use of antimicrobials, to remove those, just simply removing the antimicrobials does not make those resistance genes exit the population. They're still present, although they're in lower levels. Then the final area I wanted to talk about, I'll go through quickly, is just about some of the work that we've done using transcriptomics, metatranscriptomics, genomics in the study of E. coli 157 or supershedding in cattle. So there's a number of different factors that could account for the supershedding phenomenon in cattle, which is where cattle shed over 100,000 CFUs of E. coli 157 per gram of feces. Maybe due to the natures of the situation or the characteristics of the bug itself, different isolates of E. coli 157 or different strains may respond differently to become supershedders. It could be due to the host itself as well, and that some hosts are susceptible to becoming supershedders while others are not. And it could be due to environmental factors as well as to how well certain strains of E. coli 157 persist in the environment and lend themselves to be reacquired and infect additional animals as they enter the feedlot. So we've published quite a few papers around this area of study. One is the comparative genomic analysis of E. coli 157 isolated from supershedders and low shedder cattle. So we did a mass sampling from several thousand head of cattle identifying supershedders. We identified a total of 289 supershedders, of which and 495 low shedders. We then took those isolates, ran them through a number of screening methodologies, including antimicrobial susceptibility testing, lineage typing, pulse field gel electrophoresis, and biolog for biochemical characteristics. And from that, then we selected a small subset of isolates for whole genome sequencing of both low shedding and super shedding isolates. So this just illustrates that. This is back in the days when we were still using 454 sequencing. At that point, sequencing was still quite expensive, and so we only did 10 isolates. Now we're doing studies with isolates where we are sequencing 100 or more isolates in the studies we're doing today, relative to 10 back then. But when we did that, we sequenced both the low shedders as well as the super shedder isolates, and you can see that some of these animals were shedding E. coli 157 at greater than 10 to the 8th per gram of feces. They also differed in lineage typing and antimicrobial profiles. So based on those characteristics, we selected these isolates for sequencing to determine in a comparative genomics study, do we see any differences in the genomic characteristics of super shedder isolates relative to low shedder isolates. And this just shows the phylogenetic tree generated as a result of that. And the conclusion you can see here is that both the supershedding and low shedding isolates tended to be interdispersed in the tree that we examined. So the clinical isolates that were being isolated from humans were similar to what we're finding in the supershedder isolates and in the low shedder isolates. There were some exceptions. There was one bovine lineage isolate that we found that was specific with a bovine lineage genomically very different from that of the clinical isolates that were associated with the other lineage. So no basic differences between the low shedding and super shedding. Nothing is jumping out. Same when we did a pangenomic analysis, we find the same pattern where the non-shedders or low shedding isolates and the super shedder isolates are not clustering separately, they're intermingled. So in this study we concluded that there was no clear obvious factor that we could identify within the genome that would allow an isolate to become a super shedding isolate. But what we didn't study is we didn't study transcriptomics, we didn't study gene expression. And that could be the difference between low shedding and super shedding isolates. They may have the same genomic characteristics, but the gene expression may differ between those isolates. That's exceedingly difficult to study because if you take the isolate into the laboratory and you look at its expression on plates or in culture or even in tissue culture, those types of expressions may be quite different than the expression you would encounter in the animal. So we don't have a good model to really look at expression under the conditions that would be the same as what you would find in the animal itself. We looked at a whole bunch of factors that have been identified or proposed, whether the types of STX genes that are present, Phage type, what the Q gene alleles. All of those factors were in common between non-shedders and super shedders, with the exception of that one bovine strain that was clearly different than the rest. When we looked at that isolate, that lineage was unique in terms of being different from the clinical isolates, and other people had identified that as well. We looked at that in additional analysis. So this just shows the first number you see here is the super shedder level at the time that we isolated from the animal. So all those animals were shedding more than 100,000 CFUs per gram on day 0. It took us 4 days to get them to our research feedlot. By that point in time, only 2 animals in day 4 came close to being super shedders again. So they were super shedders on the day that we sampled them, but when we transported them to the feedlot, many of them were no longer super shedders after that period of time. And when we looked at some of the animals that were shedding the longest, for example, 342, you can see that when we looked at those particular isolates and characterized them further, the isolate that was shed on day 23 was the bovine lineage isolate. The isolate that was shed on day 30 was the clinical isolate. So these animals are harboring mixtures of E. coli 157 populations, which consist of both clinical and bovine isolates. It does look like at the super shedding state, the isolates are more likely to be clinical isolates than bovine isolates. But when you sample animals, there's going to be obviously a certain degree of chance as to which type of isolate you're going to get off of a plate when you do that sampling. So that's something we always struggle with when we're dealing with looking at pathogens, is are we getting these strains that are truly the virulent strains responsible for the pathogenic trait in the human or in the animal, or are these some of these other basically the same serotype but not the same clinical risk that's associated with those isolates. So that anomaly that we found was that all other isolates were super shedder or low shedder displayed these clinical-like characteristics except for that one isolate. It can be reasoned that those clinical-like strains may be present in the host at any time in low numbers. So basically we concluded that any animal has the potential to become a super shedder, and that those factors that are determining whether an animal becomes a super shed is not solely controlled by the genomic nature of the bug itself. So other factors may be playing a role in their colonization to reaching super shedding status. So we started to look at those factors, what some of those factors could be. One of them would be, what about the impact of the intestinal microbiome itself? Is that microbiome or the nature of the microbiome influencing the ability of the animal to become a super shedder? When we looked at that comparison between super shedders and low shedders, we found that super shedders had a higher richness and diversity of bacterial populations at the rectal anal junction than did low shedders. suggesting that the nature of the community is influencing the likelihood of the animal becoming a super shedder. So the companion isolates that might be present in the digestive tract forming that microbiome could influence the likelihood of an animal becoming a super shedder. We looked at other locations within the digestive tract, both upstream and downstream from the rectoanal junction, and we found basically the same conclusion. The level of diversity of the population You guys can hear me again? Good, I'll carry on. At least this time I realized I fell off. So basically we found higher levels of diversity and richness with the super shedder isolates, or super shedder animals. What about then interactions between the hindgut and the microbiome? When in this case we're looking at specifically the microbial population that's colonizing the intestinal epithelia. The other data that I've shared with you are data of the lumen, so it's the contents within the intestinal tract, not the population that's directly colonizing the intestinal epithelia. When we looked at the intestinal epithelia, we also found differences in the populations between super shedders and low shedders. And that could have implications because there's differential expression between those populations in the genes that would be expressed by the bacterial population, which could then influence the nature of the communication and the expression within the intestinal epithelial cells themselves. So then, after that, we've basically, up to this point, talked about the microbiome and the bacterial isolates themselves and the impact that they have on the likelihood of becoming super shedders. What about the nature of the host? Because it's all about the communication between the host and the bacteria of interest. And so in this case, what we're doing here is comparative transcriptomic analysis of expression of genes within the rectal tissue itself. We know that the rectal anal junction is the major site for colonization by E. coli 157. And when we look at the types of gene expression, we see similar numbers of genes expressed at that site, regardless if the animal's a low shedder or a super shedder. And when we looked at the differential expression, where we found downregulation of the gene expression in super shedders, there were genes that were related to immune status within the intestinal epithelium. So at that point, it looked like downregulation of those immune statuses, genes associated with immunity, may be what makes the animal susceptible to becoming a super shedder. And those included cells, genes associated with T cells, B cells, neutrophils, dendritic cells, NK cells. All of those had genes that were downregulated in the super shedders as compared to the non-shedders or low shedders. And then finally, we also looked at other regions within the intestinal tract for that gene expression. We also found differential expression in other regions of the intestinal tract, including the duodenum and descending colon. Some of those were related to immune factors. Others were also related to cholesterol metabolism. It might link in with hormonal expression and the impact that that may have on basically gene expression within the intestinal epithelia as well. So what are our conclusions then from that work as a whole? One is the potential mechanisms of super shedders. We're pretty convinced now that it has something to do with the potential alteration in the immune function in super shedding animals. What is the causative factor for that? Well, it could be from two perspectives. One may be that the super shedders themselves exhibit a state of immune compromisation themselves. So due to stress or other factors, they become immune compromised, and as a result, then they become more susceptible to colonization and the establishment of supershedding populations. Or it could be that the colonization by E. coli 157 in itself releases signaling factors to the intestinal epithelium that directly suppresses the immune system in the intestinal epithelia, and thereby allowing the supershedders to establish. We don't know which of those two possibilities it is, but we do seem to be generating evidence that there is a factor in terms of the immunity of the animal at the rectoanal junction that gives rise to the risk of likely becoming supershedders or not. And then finally, a theory as to why do we see that intermittent nature of supershedding, where we see an animal's a supershedder in the morning but not in the afternoon. We theorize that it has to do with the release of the biofilms from the rectoanal junction. So once you have a mature biofilm, you can have, it's a natural characteristics of biofilms to have large portions of it slough off in the hope of colonizing downstream environments. So in this case, when we sample an animal, if we happen to get a sample of in which one of these sloughing events has occurred from the biofilm attaching to the epithelium, then we get sufficient numbers of isolates within that sample to designate it as being a supershedder exceeding that 10 to the 5th level. If the isolates are not occurring or originating from the sloughing of the biofilm, just random release of a few isolates, then you end up getting a positive animal that doesn't meet the definition of a supershedder. So that's why then the isolates, depending upon whether there's a few being released for the biofilm or a lot, you could have the same isolate being identified in a low shedder relative to a supershedder. And that may be another reason why we didn't find any difference in our genomic comparison. So what is the factor then that allows to the establishment of these biofilms, and what is the factor that leads to the sloughing off, leading to the supershedder status? There was some theory, you probably heard about the vaccine that was approved and licensed in Canada for a period of time, developed by BioNiche to vaccinate cattle against E. coli 157. There was some concern that the use of that vaccine, if we're talking about this kind of situation where we have biofilms forming at the rectoanal junction, that if you vaccinated those animals, you could actually have a temporary increase in the amount of shedding or supershedders as opposed to non-vaccinated animals, because you're introducing basically a pressure that promotes the release or sloughing of that biofilm from the rectoanal junction. So overall conclusions, basically genomic, transcriptomic, metagenomic, metatranscriptomics enables pathogens to be characterized at a level of detail that was previously impossible. These methodologies are going to become the standard foodborne outbreak for source tracking and identification of linkages of pathogens back to outbreak points of origin. It's going to replace pulse field gel electrophoresis. That replacement's already going on, and within 5 years, we won't hear about pulse field gel electrophoresis anymore. The ability to investigate pathogen-host environment interactions. You can do transcriptomics on the host, you can do transcriptomics on the pathogen, as we've demonstrated. So ability to look at that interaction between the host and the pathogen in a manner that was previously also impossible. The informational databases that we're drawing this information are only going to prove to become more powerful. I gave the example of the gene that we discovered coding for aminoglycoside link resistance, which has been added to that database. There are labs all over the world that are doing that kind of work. Those informations will continue to flow in those databases, and they're only going to become stronger and stronger. So I think you can see from the examples that I gave that it's not too difficult to come up with a variety of different approaches, regardless of the type of pathogen of interest, of where you could apply several similar techniques to characterize the interaction of the pathogen with whichever environment it is that you're interested in, using the tools that I've described here today. So I just want to finalize by thanking my team back here in Lethbridge. I have a good group of people. We work, as you're probably aware, across a broad variety of areas. Antimicrobial resistance, E. coli 157 being one of the key areas that we're presently working in. So thank you very much.
Thank you. ¿Se oye? Thank you, Dr. Trim. First of all, we learned that maybe today or yesterday or maybe tomorrow is your birthday. Is that true? Is today your birthday? So you're celebrating already. We would like to extend our warmest congratulations. Happy birthday. ¿Sí se oye? Okay. ¿Alguna pregunta para el Dr. Tim? Any questions? I do have one, Dr. Tim, question for you.
Sure.
Have you looked at comparative genomics? We know there is a seasonal effect on the prevalence of E. coli 0157. Have you looked at comparative genomics with regard to the season, if there is a change also in metagenomics, if there is a change in the population community of microbes in the gut from this seasonal perspective?
We've looked at some of that. The overall predominating factor that determines the nature of the microbiome in the digestive tract is the diet. So diets also change seasonally, so they're confounded with seasonal effects. And so there are differences. So in our studies, diet is number one, but if you put all animals on the same diet, then you can detect differences in the microbiome between animals as well.
Okay. Do you have any plans on including in your experiments the influence of environmental factors in the comparative genomics that you're conducting with E. coli?
Looking at some of those things, so when we talk about the enterococci example I gave, we're looking in the comparative genomics there, like what are the factors within those genomes that make the isolates that we're seeing that are environmentally persistent, the ones that we see in the broader environment, what are the factors within those genomes that give them that characteristic that differs from the microbes, the enterococci that are residing either in the digestive tract of cattle or causing infections in humans. One of the key factors we want to study from the enterococci perspective, you can see how enterococci could be very effective from a source tracking perspective because the differences are so dramatic even at the species level, let alone comparing, like, you know, we can find several similar differences in E. coli if we go to the subtyping approach also, that we find that the subtypes that predominate in the cattle differ from the subtypes that predominate in the environment, which differ from the subtypes that cause infection in people. So those are very different populations, and there's something about those, their genomic nature that gives them a competitive advantage in each of those environments, which makes sense. You know, microbes are the masters of adaptation, so they adapt to the environment in which they reside. And if you remove them from that environment, they're not as competitive as bacteria that have adapted naturally to that environment that you're moving those transient organisms to. What we're interested in from the enterococci perspective, though, is the movement of those antimicrobial resistance genes. Can the antimicrobial resistance genes, because we can find Enterococcus here that is antimicrobial resistant, do they pass those resistance genes over to the Galvarium or the other enterococci that survive in the environment? And then could that potentially be a source that would pass them on to Fecalis and Faecium that then pose a threat for human health? Our work would suggest now that when you're talking about the impact of antimicrobial use on human health, the overwhelming majority of the impact that occurs that threatens human health today is the use of antimicrobials in humans in hospital and long-term care environments, not the use of antimicrobials in livestock.
Okay, thank you very much, Dr. Tim, for your time in your birthday.
Thank
We
you.
hope to have you here in person in future events.
Yeah, I'd like to have come down. And I'm sorry we had the problems with the connection.
Yeah, it was great to have you. Thank you very much.
Thanks a lot, guys. Yeah, bye-bye.